This is the micro-talk for evolution and adaptation of Fragilariopsis from the Arctic and Antarctic, sequencing new strains and investigating adaptation to rising temperatures. Our project looks at genomic differences between Arctic and Antarctic strains of the ecologically key polar diatom Fragilariopsis, and how different strains respond to a gradual rise in temperature in light of climate change and decreasing sea ice. We have performed de novo genome sequencing on 10 different strains, 5 from each pole, the majority of which have been previously identified as Fragilariopsis cylindrus. The majority of strains have been isolated in the last 5 years, whilst 1102, a model strain for F. cylindrus, has been in culture collections for over 50 years. We have also carried out an evolution experiment which involved gradually increasing the temperature from 0 to 12 degrees in 2-degree intervals over 14 months to simulate warming. Samples were taken at each temperature point to evaluate changes in the genome, transcriptome and methylome. Genomes were assembled and initially assessed using KMA analysis. Histograms of KMA spectra comparing assemblies to reads show that 1102 has a triploid signature, as previously shown by Kat Hodgkinson, whilst the other 9 strains appear to be diploid. Antarctic strains are more heterozygous. With further analysis, it will be interesting to see if strains display a high level of divergence between alleles as seen in the F. cylindrus genome paper, which uses the Antarctic strain 1102. 8 out of 10 strains have so far been sequenced with nanopore. Preliminary results comparing Illumina and nanopore reads show a high level of correspondence and coverage. We are currently in the process of scaffolding genomes with the nanopore reads before moving on to genome annotation and comparative analysis. However, we have carried out some interesting preliminary analysis between strains using the Illumina assemblies and reads. 1102 has the largest genome, as expected, due to an additional haplotype. Besides 1102, Arctic genomes are typically slightly larger. KMA spectra were compared between different strains. The largest shared coverage was seen amongst Arctic strains at 80%. Antarctic strains typically shared 50%. Both appear to share a higher degree of homozygous content compared to heterozygous content. And shared content between Arctic and Antarctic strains is much lower at around 12 to 20%. This higher level of similarity between strains from the same region is also observed when looking at average nucleotide identity to compare genomes. Both KMA-based and BLAST-based methodologies gave an identity of between 96 to 99% when comparing strains from the same region. This suggests that they are from the same species, as the interspecies cutoff is 95%. Although there is a crossover of ANI scores, Arctic strains are typically a bit higher, which fits with the trend from the KMA comparisons. However, ANI results from strains between regions are between 87 to 90%. This is lower than expected for the same species, but is higher than the value expected from samples from different species, which is below 83%. Interestingly, when comparing 18S, Arctic and Antarctic sequences overlap, but when comparing sequences from Visco and COX-1 genes, although all strains are within a clade, separately from Fragilariopsis coagulensis and other species within the same genus, Arctic strains and Antarctic strains cluster again into their own clades. So whilst the strains appear to be closely related, it is clear that they are highly diverged between the poles. Growth is monitored throughout the evolution experiment, with some strains showing a preference for colder temperatures, whilst others thrived at 4 to 6 degrees. 10 degrees appears to be the maximum temperature for the majority of the strains. For more information and growth models, visit the poster by Christina Sarkozy. Along with further comparative genomics and sequencing of different strains at different temperatures from the evolution experiment, we hope to gain an insight into how Fragilariopsis adapts and evolves in relation to warming. Thanks for visiting this poster. I'd like to thank everybody in the group who's worked on this project, as well as Bernardo at the Elim Institute who developed the KMA analysis and assembly tools. If you have any questions, don't hesitate to ask me in the breakout room.